Hey guys, today I am going to discuss you about the nucleosome solenoid model. Now the question, what is nucleosome? A nucleosome is a small part of chromosome. In past, it is called as gamma body. The word nucleosome is coined by outdate. In 1974, R. Kornberg or Roger Kornberg and Thomas published nucleosome solenoid model. Now what is nucleosome solenoid model? In this model, you can see chromatosome, which consists of three parts: nucleosome, second linker DNA, and histone 1 protein. Nucleosome consists of two parts: nucleosomal DNA, which consists of 146 base pair, and core histone protein, which consists of four types of histone protein like H2A, H2B, H3, and H4. And the total number of histone protein is 8. And in a chromosome, the total number of histone protein is 9 with added H1 protein. So the total number of nitrogen base in a chromosome is 200. The nucleosome has egg-like shape. It has 110 Armstrong diameter and 60 Armstrong height. Now the arrangement of histone proteins in core particle. If you imagine a cube, there are three opposite planes. From there, I am taking one opposite plane. I indicate those planes as number 1 and number 2. In number 1 plane, from point O, I arrange 4 histone proteins in anti-clockwise manner, one by one, like H2A, H2B, H3, and H4. Likewise, another plane from point P, those histone proteins are arranged clockwise in this manner: H2A, H2B, H3, and H4. So if you see this diagram, same proteins are placed in opposite corner of a plane. These proteins are joined together, so a steric hindrance repulsion force is produced, but those proteins are attached tightly for two regions: a stereospecific bond and another nucleoplasmin protein, which is anionic in nature, and these two factors stabilize the core histone protein part. This core particle is surrounded by nucleosomal DNA in 1 and 3/4 round and joined with H1 protein. And together all these are called chromatosome. Some nucleosomes are linked by linker DNAs and make oligonucleosome. This linker DNA is also called spacer DNA. Some oligonucleosomes make a chromatin fiber width of 10 nanometer. H1 proteins are located towards the center of oligonucleosome. For this region, interaction of H1 proteins occurs. This interaction of H1 proteins occur for H1 proteins repulsion force and the attraction force between H1 and DNA. The regions of this attraction are: DNA is negatively charged due to phosphodine and histone protein is positively charged due to arginine, histidine, and lysine. 6 nucleosomes are joined together and make a solenoid of 30 nanometer diameter. In this solenoid, there is some scaffold attachment area or SAR area. SAR is the specific area where adenine and thymine are found in plenty numbers. Scaffold proteins join in SAR region and makes a super solenoid model of diameter of 300 nanometer. This fiber is highly condensed and makes chromatid of 700 nanometer diameter. In metaphase, this chromatid is highly condensed and make a chromosome of width of 1400 nanometer. Now some extra information about histone protein. You know very well that the prokaryotic cell has no histone protein, but in E. coli cell there is HU protein which acts like H1 and H2. So this is all about nucleosome solenoid model, and next time I will be back with some useful topics of biology.